I love fish. Their biodiversity is astonishing, their behaviour often as weird as a 2-year-old on too much sugar, and the alien-like adaptations they use to survive around the world. They're also important for the economy, our diet, and many are keystone species. Unfortunately, though, freshwater ecosystems are some of the most polluted environments on the planet, and the biodiversity is struggling to cope. The understanding the mechanisms they use to survive is vital. Now, exposures don't occur in isolation in the wild. And so a main aim of my PhD is to understand how populations deal with repeated exposures, both within an organism's lifetime and across the generations, to improve risk assessments. Fish exposed repeatedly respond differently than they did to the initial exposure, and that alteration in tolerance can be due to behavioural or physiological mechanisms, or to changes in genetics and epigenetic mechanisms. My research has already shown that repeat exposures to copper shows a delayed hatching in subsequent exposures, improving the ability to survive during that sensitive development period as a physiological response. However, when you expose to mercury during that period, an increase in tolerance is seen not from a delay in hatching, but likely due to an epigenetic mechanism that alters gene regulation in fish. Epigenetic markers have even been hypothesized to be passed on to future generations of fish, like a gift of knowledge to improve survival in a changing environment. However, sometimes this gift can be more like a curse and lower survival with each generation, so research is needed. Let's talk about epigenetics for a second. Every cell in the body contains DNA code for the entire genome, like a book filled with endless combinations of the 4-letter DNA alphabet. Stem cells have access to every page in this book and can become any cell that they want, whereas skin cells, for example, have locks on most of those chapters, allowing only for skin genes and proteins to be produced, so that you don't get an eye growing out of your arm or something else weird like that. Epigenetic mechanisms are the cause for this cell specification, but that's not their only function. Like Post-it notes, they can highlight gene chapters for more or less synthesis and change the way genetic sentences are read so that there is a fast, flexible, and reversible change in gene regulation without a single change to DNA bases. My PhD aims to understand the epigenetic markers that are caused from repeat exposures throughout their life and the effect on their survival, as well as importantly identifying the extent in which epigenetic memory is passed on from one generation to another, and ensuring that aquatic biodiversity is here for tomorrow's future fish lovers everywhere.